The heart. Everything about the cardiovascular system makes sense if you remember the basic principle that a fluid is always going to move from the area of greater pressure to the area of lower pressure. That differential in pressures has to be formed in some way, and what it does in our bodies is the heart. Heart acts as a pump, just like pump in your garden is going to pump water through a hose. The heart is going to pump blood through your arteries, capillaries, and veins. The heart is a muscular organ, so the pumping is done by muscles, and we have already covered the differences between the cardiac muscle tissue and muscle cells compared to other types of muscles. It is really two pumps. There is a pump on the left side of the heart and a pump on the right side of the heart. The pump on the right side of the heart is pumping blood into the lungs to get oxygen and get rid of carbon dioxide. And then that blood comes back to the left side of the heart, to the left pump. The left pump is then pushing that blood through the arteries and capillaries and veins throughout the rest of the body, the entire body. And then that blood from the rest of the body, from the entire body, goes back into the heart and goes back into the right pump. So it can go back to the lungs. The blood circulates through those two pumps, going to the lungs, going to the body, going to the lungs, going to the body. The heart, an average heart of an average person, is going to pump about 5 liters of blood per minute at rest, much more during exercise or stress. Uh, if you remember from the blood chapter, uh, an average volume of blood in an average adult female is about 5 to 6 liters, and an average male about 6 to 7 liters. So almost all of the blood passes through the heart in any given minute, which is an important thing to remember. Uh, what uh, we are going to do in the lab is a lot of anatomy of it. You have already seen the models of the heart. So I want in this video to focus more on the physiology, but we're going to go through a little bit of basic anatomy first. Where is heart located? In the thoracic cavity, and in the middle part, which is called mediastinum. So, for instance, trachea and esophagus and heart and our aorta and big arteries and veins are found in that middle part, which is called mediastinum. Heart is contained in a sac called the pericardium. When we dissected the heart in the lab this week, we have removed most of the pericardium. Pericardium is complex. It has actually 4 different layers. And we have removed 3 of those layers. The fourth layer is the very thin, smooth covering of the heart itself, which we did not try to remove. Uh, that is epicardium. That part of pericardium that is snugly hugging the heart is called epicardium. Then the thick muscular wall is called myocardium. And then on the inside, once we opened the heart, we saw it lining all the atria and ventricles is another very thin, smooth membrane, which is called uh, endocardium. Endocardium is continuous with the endothelium, the innermost epithelium of the arteries and veins. The heart is about 2/3 to the left. The mass of the heart, about 2/3 of the mass of the heart is slightly to the left, which gives lungs a little bit more space on the right than on the left. Uh, the base of the heart is on the top, and the apex of the heart, or the point of the heart, is facing to the bottom. The way muscle fibers are oriented in the heart is not straightforward. It's not just straight up and down, but more like a spiral. If you look at a much later chapter in a textbook about development, there's a part about development of the heart, and you're going to see that as the heart develops, it starts twisting around itself and making a spiral. So every time the heart contracts, it's not just contracting very linearly, it's actually like squeezing. It's squeezing that blood out of its chambers into the arteries. Um, the the layers of the heart. We're going to do that mostly in the lab, and what we saw there. That there are two atria on the top, close to the base of the heart, and two ventricles on they're bigger on the bottom of the heart. There are differences in the thickness of the walls between the left and the right ventricle. And usually the question is posed as well, like, why is the left ventricle having a thicker wall? I think it's better to ask the question, why does the left or right ventricle have a thinner wall? The volume of blood that is pumped by the left ventricle and the right ventricle in every beat is about the same, and yet the thin wall of the right ventricle ensures that that volume of blood is pumped up under a much lower pressure, about the fifth of the pressure of the left side of the heart. And that is very, very important, because if it would have the same thickness of the wall, it would uh, push the blood under the same high pressure. What would that blood then do? It would go into the lungs, and in the capillaries of the lungs that are wrapped around very, very thin membranes of the alveoli. It would be a much higher pressure there, and the blood would exit the capillaries and enter alveoli. It would flood the alveoli with blood. We would drown in our own blood with the first breath. So that's why the left side of the heart is much thinner. The ventricle wall is much thinner on the right than on the left. The left side of the heart has to push blood with much greater pressure, 5 times bigger pressure, because it needs to push the blood through a very large, vast network of arteries and arterioles and capillaries, and then back to through the veins back. Which requires not just a great power of the muscle of the heart, but also requires that the arteries are built in a particular way. That's what we're going to cover in the next video, in order to kind of assist, aid in pushing the blood forward. Between each artery and the ventricle, there is an opening. Right between the left side of the heart and the right side of the heart, there's no opening. There's a septum. There's a muscle going in between. So there's no communication between the left side of the heart and right side of the heart. But there is communication between the atria on the top and their ventricles on the bottom. So there is a hole between the left atrium and left ventricle, and the right atrium and the right ventricle. When as the blood is going from the atria into the ventricles, it's going from high to low pressure as the blood is coming in from the veins, and ventricles are relaxed. When the heart beats, when the ventricles contract, that increases the pressure in the ventricle. The blood is going to go down its pressure gradient. One way to go is into the arteries like the aorta, but there's another way to go, and that is back into the atrium. So this is why there are valves in the heart. On the left side of the heart, there's a bicuspid valve or mitral valve, and on the right side of the heart is the tricuspid valve. When the heart contracts, and that blood under high pressure tries to push back into the atria. Those valves close. Remember that the valves are made of fibrous tissue that is connected via cords, chorda tendinae, that act like little tendons uh, to the papillary muscles. Papillary muscles are part of the heart muscle, so they're going to contract at the same time when the rest of the ventricle is contracting, tightening and tightly closing those valves so they don't open under the high pressure coming from the ventricles. There are also two other valves, and that is the valves between the ventricle and its artery. So on the left side of the heart, that is going to be a semilunar valve between the left ventricle and the aorta. On the right side of the heart, that is between the right ventricle and the pulmonary artery or pulmonary trunk. Uh, that's another semilunar ventricle. So the pressure of the blood during the contraction of the heart is going to open the semilunar valves and allow the blood to go into those arteries. When the ventricles then relax, the pressure in the ventricles goes down, while the pressure in those arteries is still high. They're full of blood right now, so there will be uh, a force to push the blood back into the ventricles. This is what closes the semilunar valves, so the blood cannot go back into the heart, has to keep going on away from the heart through those large arteries. And we have seen the chorda tendinae and we have seen the valves when we dissected the heart in the lab. What there are a couple of other things to note. One is that when one day later in this semester, when we dissect a fetal pig, we're going to see red arteries and blue veins. That's not natural. They have been infused with latex in order to be able to see them better. Um, there it's actually they are not colored in a fresh or any kind of preserved specimen, either human or pig or cat, whatever you're dissecting, unless there's latex infused into them. So I actually sent by email and also put in additional readings a link to how to differentiate arteries from nerves when you're dissecting a body that has not been infused with latex. Uh, arteries, the word that's used for all the blood vessels that are taking the blood away from the heart. And they are in all the pictures, all the images, lab manuals, atlases, they are always painted red. That's kind of a universal code that we have agreed on to always label arteries in red. Veins are always labeled in blue. Veins are all the vessels that bring the blood back into the blood, into the heart. Now, that is different from which blood is oxygenated and which blood is unoxygenated or deoxygenated. Remember that the blood gets oxygenated inside of the lungs. This means that the blood that is going from the heart to the lungs is deoxygenated. It's going to the lungs to get the oxygen. When it's going there, how? By pulmonary arteries. Pulmonary arteries are going from the heart to the lung. Although it's an artery, it is not carrying oxygenated blood, it's carrying deoxygenated blood. Once it picks up oxygen in the lungs, it's going back to the heart via pulmonary veins, and yet that is oxygenated blood. So it's not synonymous artery always oxygenated and vein always deoxygenated, because in the pulmonary circulation it's the other way around, deoxygenated in arteries and oxygenated in veins. Everything else, so blood coming out of the left side of the heart and going to the rest of the body, all of the body, starting with the aorta, is going to carry oxygenated blood in arteries, and it's going to return from the tissues deoxygenated blood into the heart via veins. So you have to separate those two divisions. That the arteries and veins are one thing, oxygenated and deoxygenated blood are the other thing, and they don't always go together. Um, Blood is beating all the time, which means it's taking up a lot of energy. We talked about muscles, how much energy they take, a lot of ATP, and and yet the blood itself cannot, when it's inside of the heart, it is separated from the heart muscle by the endocardium. So it cannot provide oxygen and nutrients to the heart muscles from within, which is why the first two tiny little arteries coming off of aorta as soon as it leaves the left ventricle are the two main coronary arteries. Coronary arteries then divide. You can look in your textbook exactly what are the names of all those main divisions, and they are providing blood to the heart muscle itself. And they are uh, very efficient. They're bringing, they're releasing a lot of oxygen into the heart muscle at all times. And likewise, their coronary veins that are bringing back that blood back into the heart. So uh, uh, coronary circulation is the circulation of a number of small arteries and veins that are providing oxygen and nutrition to the heart muscle itself. Because the heart is so uh, oxygen hungry and energy hungry, because it's working so hard, it's a big muscle that works all the time. uh, it has to protect itself from the possibility that some of those tiny little skinny coronary arteries might get clogged or something goes wrong with them. So it has redundancy in it. What does that mean? That there are arteries that cross from artery to artery, which means there's more than one way to bring blood to any part of the heart. Those artery to artery crosses are called anastomosis. And an anastomosis uh, is a direct link from artery to artery or from artery to vein without going through any kind of capillary beds. Uh, so that's how the heart is ensured to always have oxygen and glucose, by making sure that the blood supply can get to every part of the heart by more than one way, just in case. Now, if it gets really, really clogged, some of those redundant ways are also going to get clogged, and then you have a heart attack, you have infarction of the heart. Uh, the cardiac muscle, as we have already discussed when we were talking about muscles in general, are somewhat different from the skeletal muscle. Skeletal muscle are very long and thin and very long, and each muscle cell is the length of the entire muscle. Skeletal muscle cells don't branch. Skeletal muscle cells have dozens, sometimes hundreds nuclei. Cardiac muscle, as you remember, uh, doesn't get that long. The muscle cells don't get that long. They do branch and connect with each other, and they only have one, two, maybe three sometimes nuclei. Um, so cardiac muscle cells produce a net because they're connecting, branching and connecting with each other. They produce a net. They're connected with each other. They're connected with each other with membranes called intercalated discs, which you have seen on the slides in the lab because they were dyed on those preparations. You could see them as little black lines. What is important, and already talked about that before, is that those membranes have gap junctions. Gap junctions allow for direct or electrical signaling, which is the fastest type of signaling, which means that the ions can go directly from one cell to the next. When are ions going to go from one cell to the next is when the first cell is undergoing its own action potential. That action potential can that way move from one cell to the next without having to wait for any kind of synapse or anything like that. Well, that ensures that all muscle cells in the heart are contracting essentially simultaneously. The whole heart beats all at the same time. So atria do it all at the same time, and there's a little delay, and then the ventricles all contract at the same time. Uh, so that is important to know about the cardiac muscle. Um, the cardiac muscles cells, cardiac muscle as a tissue. So the heart beats, and the heart beats in a dish. If you take the heart out of a body, it will continue beating for a while. If you provide it with, all the nutrients it has, it can beat in the dish for quite a while. How does it do? So it doesn't require, let's say, a nerve impulse to constantly tell it beat now, beat now, beat now, beat now. The heart does it itself. It has an autonomous pacemaker. There are muscle cells in the heart that have been modified, and they're capable of producing their own spontaneous action potentials. The action potentials of the cardiac muscle are different in some important ways from the action potentials in the neuron or the action potentials in the skeletal muscle, which is always induced by acetylcholine coming from a motor neuron. Uh, Action potentials in cardiac muscles are different because they involve different types of ion channels. Remember, in the axon, the action potential starts with opening of the sodium channels, and once all sodium channels are open and it's maximally depolarized, that opens all the potassium channels, which leads to repolarization. That is the same thing is going to happen in a skeletal muscle that has been triggered. Cardiac muscle is different. It has different types of sodium channels. Those are leaky channels. There's a slow leaky channel. Sodium is constantly leaking in and bringing the membrane slightly more and more positive, depolarizing spontaneously on its own. And when it does, reach a threshold, it is going to trigger opening of calcium channels. Remember, there's no calcium channels at the axon hillock. Cardiac muscle has calcium channels right there. They're going to open, and they're going to stay open for a long time. They're going to stay open anywhere between 100 and 250 times longer than an action potential in a neuron. It's going to be a long, long time that the action potential in the cardiac muscle exists. Instead of 2 microseconds, it's going to be 200, 300, 500, not microseconds, milliseconds. Um, and uh, during that long time that the calcium channels are open, the muscle is refractory. It cannot start another action potential. It's contracting. It needs time to relax before it contracts again. You don't want a tetanus of the heart. Uh, so this is a long plateau, a depolarization phase that's initiated by these leaking sodium channels is going to continue into long plateau phase. That plateau phase is there because of the calcium channels or being open and not closing very, very fast. This voltage is also going to start opening potassium channels. So potassium is going out, calcium is going in. They kind of balance each other. The voltage around the membrane is going to stay the same for a while until the calcium channel starts closing. Then it's the potassium movement that is driving the membrane potential, which is going to lead to repolarization, going back to more negative on the inside of the cell, after which there's no more refractory period and the heart can beat again. So it's a much, much longer action potential than in the axon or in the skeletal muscle. And it is because calcium channels are there, and calcium channels are open for a long time. The heart can do it on its own without needing an input from the outside because its sodium channels are leaky channels. They are always allowing some sodium into the cell. So that is the important difference between the action potential and the cardiac muscle cells compared to the skeletal muscle and the neuron. Remember that not all cardiac muscle cells are like this. Only the cells of what is called a conduction system of the heart. Conductive cardiac muscle cells are specialized this way. The other muscle cells act more or less like skeletal muscle cells. They need to be triggered. They need to get those ions coming from the other cardiac muscle cells. The conduction system of the heart needs to send the action potential to the other cardiac muscle cells, the contractile ones. So the conduction system of the heart is actually quite complex. It has several different layers or several different centers. The main pacemaker of the heart is called the sinoatrial node or SA node, and it's found in the right atrium. This is where it all starts. Once those cells in the SA node have their own action potential and contract, they're going to send impulses by various fibers to other different places, including to the left atrium. Thus, the left and right atrium are going to contract about simultaneously. Some of the fibers are going to go from the SA node to the atrioventricular node or AV node. The AV node is embedded in the skeleton, a fibrous skeleton that lies between the atria and the ventricle. There's the same skeleton that contains the uh, the valves, the mitral and the tricuspid valve. Part of that skeleton is the AV node. AV node takes a while. AV node produces a delay. That delay is very, very important because the atria need to finish contraction, pushing the blood into the ventricles before the ventricles contract, pushing the blood out into the arteries. You cannot have those going simultaneously, which is why AV node is important to engender that delay. Uh, AV node then continues or pushes the impulses into what is called an AV bundle or bundle of His. I prefer the bundle of His because wherever you look, wherever you read, from Wikipedia to textbooks to atlases, it's going to say the bundle of His. So I would like you to know that atrioventricular bundle is this kind of newfangled translation. They want to erase history by removing the names of people who discovered those structures, although everybody uses those names and not the new names. Uh, Bundle of His then separates into the left and right bundle branches, and then bundle branches go all the way to the apex and then branch into what is called Purkinje fibers. So all of that is going through the septum between the two ventricles. It's not really uh, resulting in any kind of muscle contraction as it's going down. It goes to the apex, and then the Purkinje fibers are interspersed throughout the whole muscle tissue of the ventricles, and it is going to be telling the ventricles to contract. So SA node is going to tell the atria to contract, and they're going to contract from the top down, while the AV node is going to send the impulses to the apex first via a bundle of His in the left and right bundle branches, and is going to start initiating the contraction from the apex up. So the atria contract from the top down, the ventricles contract from the bottom up, which makes sense because they need to push the blood into the arteries that are on the top. If you take these parts, different parts of the conductive system of the heart, if you put them in a dish and give them, medium with nutrients and everything like that, they're going to beat in the dish, and they're going to beat at different frequencies. The sinoatrial node, the SA node, has the highest intrinsic frequency, which is up to 100, beats per minute in a dish. And then the next one is AV node, which is a little bit slower in the dish, and the bundle of His is slower still. And then the left and right bundle branches also contract in a dish, but even slower. And the Purkinje fibers also contract, but only a few beats per minute, very, very slowly. What that means is if there's something happens to the SA node, the AV node then is going to take over as a pacemaker. The atria are going to have trouble contracting, but the ventricles will contract much slower, but they will keep beating. If something happens to the AV node, the bundle of His is going to initiate contraction of the ventricles. The heart will continue beating much, much slower, but it will continue beating, which means you might be able to survive. And the same thing, if something happens to the bundle of His, the left and right bundle branches are going to continue driving the beating of the heart. And the Purkinje fibers on their own can drive the beating of the heart. Of course, if the heart is beating 5 or 6 times a minute, you're probably going to faint and die, but at least it can work. So if I give you a bunch of numbers at the exam and you say, what do you think is the intrinsic, genetically programmed frequency of each one of those parts, you just remember in which order they go. SA is a pacemaker because it's the fastest, it's going to drive the rhythm of all the others. AV node is next, bundle of His is next, the branches are next, the Purkinje fibers are last, are going to have the smallest frequency of spontaneous contraction. Okay, then all right, now we go to EKG. EKG is a measure of those action potentials, right? There's electricity happening there. There are ions going in and out of the cells, and that is flow of current. That flow of current can be detected by electrodes of the EKG apparatus. And if you put the electrodes on yourself and start measuring, you're going to see a very specific shape of the wave. And different parts of that shape have different names. There's a P wave, then there's a QRS complex, and then there's a T wave. And what is going to happen is that you will learn what each one of those means. So P wave is depolarization of the atria. Depolarization of the atria. That means the beginning of the action potential of the atria. And then the QRS complex has actually individual waves, QR and S, but they are all covering each other. And that results from depolarization of the ventricles, and which is just before the ventricles start contracting. The T wave represents repolarization of the ventricles. So when they repolarize, the potassium is going on. You cannot see repolarization of the atria because they are masked, they're covered by the QRS complex. So uh, we can look at various different types of EKGs and from there figure out if there's anything wrong with the conductive system of the heart. For instance, if you have QRS and T waves going nicely, but P waves sometimes happen, sometimes doesn't. So you have a P, QRST, P, QRST, and then no P, just QRST, QRST, P, QRST, P, QRST. So some P waves are there, some P waves are not there, but QRST goes just fine. What is that? That is the atrioventricular block, not primary, not tertiary, it's secondary. It's the second order, second degree block, which means that sometimes the impulses uh, from uh, the the atrium go through the AV node. Sometimes they don't. Anyway, uh, this brings us to the whole cardiac cycle. You can look at EKG in order to figure out what is happening electrically. You can listen to the sounds, and there's a whole video about the Kortkov sounds and how you figure out when each one of the valves is opening and closing. And then of course there's a movement of blood through the heart. So you would need to put those together. They're wonderful pictures and videos and animations in your textbook showing you what is simultaneous with what when you're looking at EKG, when you're looking at blood flow, when you're looking at sounds, when you're looking at pressures of blood in various parts of the heart. Uh, the heart sounds, as I said in the uh, other video, are due to the turbulent flow of blood during the period when the valves are closing, so narrowing the opening. When they're completely open, it's laminar flow. When they're completely closed, there's no flow. When they're in between somewhere, they are going to be audible because there's turbulent flow and blood cells and proteins and all those things are hitting each other, hitting the walls, and producing sound. The instrument we use for detecting sound is stethoscope, which we're going to use to listen to the heart. Um, there are some important measures of blood, and probably the most important is cardiac output. That's the volume of blood pumped by a ventricle, one of the ventricles, either one of them, each minute. And it can be calculated. Stroke volume is the volume of blood pumped per ventricle each time for each contraction, and the heart rate is the number of contractions per minute. So if you multiply those two, stroke volume and heart rate, how many times the heart beats per minute, and how much blood it ejects each time it beats, you get the total volume of blood pumped out by that ventricle during 1 minute, which is the cardiac output. So that cardiac output is about 5 liters per minute. Remember, we said it at the very beginning that the heart beats at rest about 5 liters per minute. Uh, you can calculate very easily using this formula that you have in the book the number of beats, the stroke volume being about 70 milliliters per beat. Let's say about 70 beats per minute, you end up with about 5 liters per minute. This is an average person at rest. Athletes tend to have lower heart rate because they have a higher stroke volume, because the heart is bigger and stronger and it can push more volume of blood per stroke, per contraction, which means that it doesn't need to beat as much in order to get the same cardiac output. So 60 is totally normal for a top athlete, uh, beats per minute. When when the when we start exercising, so we exert uh, more demand on the heart. The heart is going to beat faster, and it is going to uh, maybe increase the stroke volume a little bit. But as it beats faster and faster, so you're going to have more beats per minute, and you even if you don't increase the volume per stroke, you're going to increase the total cardiac output. There's a limit to that. Once you get to about 120 beats per minute, uh, that increase of cardiac output stops. And as you keep increasing the heart rate, it's actually the cardiac output actually starts going down. Why is that so? So you're increasing the heart rate, and yet cardiac output is going down. You just plugged it into the whole formula. What is happening? Something's wrong with the stroke volume. The stroke volume, instead of going up or staying the same, is going down. Why? Because when your heart is beating that fast, it cannot refill the ventricles fast enough. The blood coming from the veins is not bringing enough blood into the atria, and from the atria to the ventricles. And so the stroke volume, the volume that is ejected in one beat, gets lower, resulting in a lower cardiac output once you go about 120 30 beats per minute. Okay, the regulation of the heart. So, we already talked about the ability of the heart or different parts of the heart to beat in a dish, which means that the heart can regulate itself to some extent. It does not need, for its proper function, to get impulses from the controlling systems. Remember how we talked about sensory nervous, circadian, and endocrine system as controlling systems, the smart systems that can evaluate the environment and make decisions and tell all the other systems what to do. Well, those other systems usually know how to do only one thing. Heart only knows how to beat. But a lot of other organs and systems have some degree of autonomy. Remember that the gastrointestinal system has its own nervous system, right? Heart actually has its own nervous system a little bit too. And heart secretes some hormones, and heart can regulate some of its heartbeat on its own without getting any kind of impulses from the outside. Uh, so, for instance, the power of the heart, the strength of the contraction is going to increase if the heart is stretched more. How do you stretch the heart more? By filling it with more blood, right? So if there's more blood in the atrium ventricles, it's going to stretch the muscles more, and more the muscle is stretched, with greater force is going to contract. That is called the Frank Starling's law of the heart, which is nice to know, but it doesn't really concern us as far as uh, normal situation, because Frank Starling's law has a limit. Remember how actin and myosin make these cross bridges, and so more you stretch the muscle, fewer the cross bridge is going. There's going to be a greater strength of contraction as the two filaments scoop back all over each other. But there's a limit. If the actin and myosin are completely separated, not a single head, they can't contract anymore at all. Um, so Frank Starling law has a limit. But this is never going to happen in a heart, because heart is enclosed in the pericardium, there are lungs on the side, there's the diaphragm on the bottom, there are ribs around it. So it's never going to stretch that much. It could potentially during open heart surgery. So this is something that surgeons know to control for. Um, as much as heart can kind of regulate its own beating and its own contractions, it also needs to be responsive to the environment. It can't really uh, sense the environment on its own. It has to get impulses from one of those controlling systems, the sensory nervous circadian or um, or endocrine system. It needs to get inputs from those systems in order to speed up or slow down the heart rate. Uh, it's going to respond to impulses that are coming from a sensor being integrated and then telling the heart what to do. There are different types of sensors that directly or indirectly can affect the heart rate. One is the baroreceptor. Those are stretch receptors that monitor blood pressure in the aorta and internal carotid arteries. They are going to affect the medulla oblongata, which has a cardioregulatory center, which tells the heart to speed up or slow down. How does it do that? Using the sympathetic and parasympathetic fibers. Now, remember that the normal heart rate resting is 60 70 beats per minute. Remember that if you put the SA node in a dish, it's not 60 70, it's 100, 110, 120 beats per minute. So the pacemaker would try to tell the heart to beat twice as fast as it would normally beat. How does that happen? It is because the cardiac center in medulla oblongata is constantly telling the heart to slow down using the parasympathetic fibers. And then, if there's a need to speed up the heart, the first thing the medulla oblongata center does is slightly release that parasympathetic input. And so the heart beats up. Once it gets to a certain level, it actually adds the sympathetic input to speed it up even more. Uh, one one of the situations when medulla oblongata would do that, that center would do that, is if it gets inputs from the baroreceptors, the stretch receptors that are measuring blood pressure in the aorta and in internal carotid arteries, telling you that the blood pressure is low. You increase blood pressure by increasing cardiac output, which you can do by increasing the heart rate. Uh, the Another one is chemoreceptors. So uh, there are chemoreceptors in various places, including in the medulla oblongata itself, that are going to uh, monitor changes in oxygen, changes in carbon dioxide levels, and changes in pH of blood. And so if pH drops or oxygen drops or carbon dioxide is too high, these chemoreceptors are again going to act on the cardiac center in the medulla oblongata to speed up the heart by really removing or inhibiting the parasympathetic input and increasing the sympathetic input into the heart. Um, so baroreceptors and chemoreceptors are going to affect the heart rate. There are also hormones that are going to affect the heart rate, and you already know it: adrenaline or epinephrine and norepinephrine secreted from the adrenal medulla are going to speed up the heart rate. When is it most likely to happen? During stress, when you're a zebra being charged by a lion. Okay. And this is already a very long video, so I'm going to stop now. And tomorrow or Saturday, I'm going to make a video on the blood vessels.